Okay, welcome to our first DNA video. This could be mostly a review of information we already went over during biomolecules. But while we're doing, I want you to take your Cornell notes and think about the essential question of how does DNA make each of us different? Now I'm going to go pretty fast, so if I'm going too fast, just pause and rewind as needed. Right, so let's start with what DNA is. So DNA is deoxyribonucleic acid. Deoxyribo just means the type of sugar that makes up our nucleotide. Nucleic, you can remember, since our DNA is found in the nucleus. So the D is deoxyribonucleic acid. That's what it is. There's a nice picture of it. And it is the blueprint of life. So it gives the instructions for how our bodies do things. And DNA is a polymer, so it is made up of a whole bunch of monomers, and the monomers are going to be nucleotides. And all organisms have lots of them, between thousands or even millions of them. In fact, our bodies, we have up to approximately 250 million base pairs long in some of our DNA. So you can see in this picture here I have a nucleotide, and I actually have 12 of them, and that they have come together to form a strand of DNA. So let's look closer at that nucleotide. So this is our pentose sugar, so it's a 5-carbon sugar, so it's a CHO. It is a nitrogenous base over here. So this is where I add my nitrogen, my N, and I have a P or a phosphate group. So I end up having CHOMP. We learn this as our penthouse. We call it a penthouse because it has 5 sides. With our basement, our nitrogenous base, and our swimming pool. So our house with our basement and our swimming pool. And if you remember, we have 4 possible bases. We have adenine, thymine, cytosine, or guanine. So this base right here is going to either be an adenine, thymine, cytosine, or guanine if we're talking about DNA. We like to abbreviate it with the first levels, or the first letter. So A is adenine, T is thymine, C is cytosine, G is guanine. Nice little diagram, so you can see here how our phosphate group and our sugars are all bonded together. That forms our backbone. And then inside we're going to have our nitrogenous bases. So we have our T, this is a C, a G, and an A. So this is how they come together. So we have our backbones. So we have a backbone on both sides because this is like that ladder, that twisted ladder. So these are the sides of the ladders here. But you can see that the phosphate group binds with the pentose sugar, binds with the phosphate group to the sugar, phosphate group sugar, phosphate group sugar, phosphate group sugar, and so on. Those are our backbones. The rungs of our ladder, or the inside of our ladder, are going to be our nitrogenous bases. And they pair with each other. So an A always pairs with a T, and a C always pairs with a G. So on this far left, we have an A paired with a T. Then we have a G with a C, T to A to T, C to G, G to C. They were always going to pair with each other. The other thing you're going to notice in this diagram are going to be these dotted lines between our nitrogenous bases. So we have 1, 2, 3 dotted lines between that C and that G. That's just showing that the C and the G pairs with each other, bonds together with a hydrogen bond. If you remember, hydrogen bonds are not really strong bonds, but there are 3 hydrogen bonds between a cytosine and a guanine, while there are 2 hydrogen bonds between a thymine and an adenine. Now, these pictures are the pictures of the people that discovered that structure of DNA. And the first two, Watson and Crick, are pretty much considered the ones that figured out that structure. In fact, if you ever watched Miles from Tomorrowland, they are honored in there. This is a character from that TV show, and it is Admirals Watson and Crick. So these two guys that are stuck together is that's an admiral on that cartoon, and they are honoring the two people that discovered the structure of DNA. I want to point out this last one, Rosalind Franklin. It was her work that really allowed these other two guys to figure out that structure. And she was really dedicated to her work. In fact, she was so dedicated to work that it killed her. So she took x-rays, and this bottom right picture is an x-ray that she took. And because she was exposed to all those x-rays, she ended up getting cancer and died. Unfortunately, she died before they handed out the Nobel Prize, because she would have gotten a Nobel Prize along with the other guys for discovering the structure of DNA, and that it was in fact a double helix. Another guy you're going to probably hear about is this guy, Chargaff, and you're going to hear about him when we're talking about how our A's and our T's pair together and our C's and our G's pair together. He did some research and he noticed that if you put two of them together, they would have been too big and the other two would have been too small. So he figured out that an A and a T had to go together because they would be the right size. And if there was, I don't know, 30% A, then there would also be about 30% T. So he's able to use some math and figure out that A and T go together and C and G go together. And you're going to hear that as Chargaff's rule. And because of his rule, we're able to do some calculations here and figure out how many or what percentage of the rest of the nitrogenous bases there are. So let's start up here in the top left. So if I have 30 A's, I am always going to have 30 T's. And I like to keep my math simple, so I'm going to split it in half. So I know I have to have 50 on one side and 50 on the other side. So 50 minus 30 is going to be 20. So I'll have 20 G's, which means I have 20 C's. And we could quickly do some math to make sure that we are right. So 40 plus 60 is 100, so I got 100% of them. Or 50 plus 50 is 100, I got 100% of them. So on the top right one, I have 5 T's, which means I have 5 A's. And if I do my math, 50 minus 5 is going to be 45. So I'll have 45 G's, which means I'll have 45 C's. And I can do my math, 90 plus 10 equals 100, or 50 plus 50 equals 100. So the bottom left one, I have 15 C's. That means I have 15 G's. And do my math again. So 50 minus 15 is going to be 35. So I'll have 35 A's, 35 T's, and I could make sure I'm right. 50 and 50 is 100, or 70 and 30 is 100. And bottom right, 40 C's, 40 G's. So if I do my math, 50 minus 40, I'll have 10 T's, which means I'll also have 10 A's. So I have 20 plus 80, that's 100, so I know I'm right. So you'll get some more practice doing that in the future. But these order of nucleotides, that A, T, C, G, are really important because that's what make all of us different. In fact, that's what make us different from other living organisms. That's because the sequence of our DNA determines our RNA, and that sequence of RNA determines what protein is made. So that central dogma of life, DNA makes RNA, RNA makes protein, and it's the sequence of nitrogenous bases that's important. That's it for this video. Next video we're going to talk about chromosomes, chromatin, some genes, and karyotypes if you want to get a head start. But I want to leave you with this slide because I thought it's pretty interesting, and this is the percentage of genes that we share with other organisms. So according to this chart, 98% of our genes are the same as a chimpanzee's genes, and 92% of ours are the same as a mouse. 84% of our genes are the same as a dog. About 80% of our genes are the same as a type of fish. 65% of our genes are the same as that chicken you'll probably eat for lunch. Half of our genes are the same as a fruit fly. About 20% of our genes are the same as a common weed that you can find in the garden. About 20% of our genes are the same as a type of worm. And between 10 and 18% of our genes are identical to genes found in bacteria. So just think about that as you move on and we start to explore our DNA a little bit more. So good luck with your DNA projects. If you have any questions, please come see me.